My name is Omar Anwar, and this is my first ever paper walkthrough of a past paper from January 2021. This is paper 1BR. Okay, so the first question is, organs in the human body have different functions. Name the organ that produces bile. Well, the first thing that we need to note is, what is bile? So bile is, first of all, bile is alkaline, and it goes into the stomach. Now, the stomach contains HCl, which is very acidic. So since bile is alkaline and it goes in the stomach, which is acidic, this will cause neutralization in the stomach. So bile neutralizes the hydrochloric acid in the stomach. Bile also emulsifies fat. Emulsifies fat means it breaks it down into smaller pieces. So when it's broken down into smaller pieces, it increases the surface area for lipase to react with the fat. So the organ that produces the bile is the liver. Now, if they ask you where is the bile stored, the bile is stored in the gallbladder. So stored. Now, obviously, the only thing you need to write for this question is liver. However, this is just further explanation and possible things that they possibly can ask you about. So the next question is, which organ releases progesterone? I believe for paper 1 biology, these are the hormones that you need to know, and ADH, FSH, and LH are all for paper 2. So progesterone is produced in the ovaries and it supports pregnancy. Progesterone maintains the lining of the uterus. So progesterone is produced in the ovary. Okay, now, which row of the table correctly shows whether the kidneys and skin are involved in excretion? This is a very easy question because kidneys produce urea, which contains salts and minerals, and skin produces sweat, which also contains ions and salts. So both of them are excretory organs. Now, the stomach is an organ found in the alimentary canal. Describe the role of the stomach. So this question has 3 marks, which means you need to state 3 things. Remember, all it's asking you is to describe, not explain. So the first thing, which is pretty easy to know, is that the stomach has a muscle lining. So this muscle lining is used for mechanical digestion. And the second point is that the stomach breaks down proteins. The third point is the stomach contains enzymes such as protease, which helps break down proteins. And also you can state that the stomach contains hydrochloric acid, which maintains acidic pH. Okay, so this diagram shows a section through a human heart with the chambers labeled P, Q, R, and S. Now, over here, you have to note that this is the right side and this is the left side. Now, obviously, when you're looking through this at a paper, this is going to be on, like, when you look at this at the paper, this side is technically your left and this side is your right. However, if you take that paper and turn it around and put it on your body, you're going to see that the right side is this side and the left side is this side, because remember this is a heart, so you're viewing this as something that is 3D. Okay, so now oxygenated blood is obviously going to enter from the pulmonary vein. Pulmonary just means the lungs, and whenever you see vein, just vein equals back, so that means it's carrying blood back to the heart. Now this color, the color red, shows that it's oxygenated because blood gets oxygenated in the lungs. Okay, it's going to come from the pulmonary vein into the left atrium and then through the bicuspid valve and from the left ventricle and out through the aorta. Remember that aorta pushes the blood away, so A equals away and then V equals back. And then if you notice this, this question could also be asked. They're usually gonna ask you, why is the left side of the heart thicker? You just need to say that the muscle needs to be thicker because it's stronger since it's pushing oxygenated blood all throughout the heart, so it needs to contract harder to build up more pressure. Okay, now on the right side, blood comes back through the vena cava into the right atrium, down the right ventricle. It's going to go up to the pulmonary artery. It's going to go to the lungs, and it's away from the heart because A for away. Now over here we have the tricuspid valve for the right side. So just say tri, RI for the right, and we have bicuspid for the left. And obviously we're going to have the semilunar valves. However, usually no questions are asked about the semilunar valve, but it's nice information to have. Okay, so which chambers carry deoxygenated blood? Remember that this is our right side, this is our left side, this is our vena cava, this is our pulmonary vein. So over here, Q, which is our left atrium, over here, the left atrium is going to carry oxygenated blood. So this area is oxygenated. This area is deoxygenated. So it's going to be P and S. Now explain why two of the valves in this heart are closed. Okay, so this is a really important question. Number one, valves are closed, okay, to prevent the backflow of blood. Okay, to prevent the backflow of blood. Our second point is it to maintain the pressure in the blood. Okay, so explain why two of the valves in the heart are closed. Number one, which is very important, to prevent backflow of blood. Number two, to maintain pressure in the ventricles. It's really important to have pressure over here so that the blood, when this muscle over here contracts, it pushes away all the blood out. Okay. Heart disease is a major risk in the heart disease is a major risk to health in the United Kingdom. In a study, the number of people with heart disease was recorded. The table shows the results of the study. Okay, so number of people with heart disease per 1,000 in a population. We have males, females, 18 to 24 are ages. Okay, males are higher over here, also higher over here, higher and higher. Okay, so give two conclusions that can be made from this study. Well, first of all, males are more at risk from females. So you can say males are at a higher risk of heart disease. And then the second one is, notice how as the age gets higher, the number also gets higher in both. So heart disease increases as age increases. Okay, the population of the United Kingdom is 65 million. Okay, so I just like to write out my numbers. 1, 2, 3, 4, 5, 6, of which half are male. So half of that. Let me get my calculator. So that is 32,500,000, of which half are male. Calculate the number of males with heart disease between the ages of 18 to 24. Okay, so since this is every 1,000 people in the population, we're going to have to divide this number by 1,000. So you have your thousands over here. 1,000 has 3 zeros, so just 1, 1, 2, 2, 3, 3. So that's going to give us 32500. This is how much males per 1,000. And then from 1,000, if we go back, okay, from 1,000, I believe it was 5 per every 1,000. So times 5 equals 162,500. Boom. So just write that here. Okay. It's very important that you write out your steps so that you can get the 2 marks. Next question. Explain how heart disease can affect a person's health. Okay. This is called atherosclerosis, which you will be covering in your A-level biology. However, I thought I would include this diagram as it's a very nice and interesting diagram. Okay, so heart disease is mainly the blockage of the coronary artery. So okay, so the blockage of the coronary artery will cause less blood to flow to the heart. So when there's less blood flowing to the heart, that means that there will be less oxygen circulating around the blood. Um, so just write less oxygen because less blood is being oxygenated. Which results in lower respiration. And then you can always say that, notice over here how when there's less blood, the muscle starts to turn into this darker brown color, just means that the muscle is dying. So you can then say that the heart will contract less, or you can say a heart attack. So we can just say um, heart muscle deteriorates, or heart muscle dies, leading to heart attack. Okay, so now they're asking about gas exchange. So alveoli are involved in gas exchange. The diagram shows a section through one alveolus and its associated blood capillary. Name the organ that contains the alveoli. Okay, this is a very simple question. The lungs. So one thing to note about the alveoli is that it is moist and that capillary is 1 cell thick. Okay, there are 5 million red blood cells per millimeter squared, cubed of blood in the capillary. Give the number of red blood cells per millimeter cubed in standard form. Okay, so this is our number 1, 2, 3, 4, 5, 6. Okay, we have 6 zeros, so that's just going to be 5 times 10 to the power of 6. Okay, explain why gas exchange is possible at the alveolus. Okay, this is a very important question. So, number one, you guys have to notice that there is a concentration gradient. This concentration gradient is caused when there is a lot of oxygen on one side, so high amounts of oxygen over here. Wait, let me make that in red. Okay, so there's a lot of O2, 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 O2. There's little O2 here. So there's going to be a concentration gradient. Note that in biology, things will move from a high concentration gradient to a low concentration gradient because this process is by diffusion and it does not require any energy. So a concentration gradient allows the diffusion of oxygen from the alveolus to the red blood cells. The red blood cells are inside of the capillary. Okay, so explain why gas exchange is possible at the alveolus. Well, a concentration gradient allows the diffusion of oxygen from the alveolus to the red blood cells. Over here, I believe you will gain 2 marks because number 1, you mentioned the concentration gradient, and then number 2, you mentioned the process of diffusion of oxygen. So this is 2 marks. And then for the 3rd mark, you can say that for the 3rd mark, you can just state that the cell is the blood capillary is 1 cell thick. And then for just for your own knowledge, you can also say the alveolus is moist. This moisture just aids in diffusion. Okay. So usually they're also going to want to see your skills on how you interpret graphs, which is a really important process in statistical analysis and in scientific evaluation. However, this will be further explored as you go into your A levels and university. So the graph shows the relationship between the total surface area of alveoli and the respiration rate in different sized animals. Okay, so our respiration rate is on our x-axis, and then total surface area is our y-axis. Okay, so we have bat, mouse, rat, guinea pig, monkey, cat, pig, dog, human, cow, whale. Oh, look at that. You have a bat. The bat is very small. So the bat are small organisms. Whales are huge. 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 I spelled that wrong. Huge. Okay, so give 2 conclusions that can be made from this graph. Oh well, look at that. Notice this increase. So, respiration rate increases as surface area increases. And then you can either say that big animals have the highest rate of respiration, or you can say small animals have the lowest rate of respiration. However, note that you cannot make these 2 points as your 2 conclusions because they're basically the same thing but reverse, so they're only going to give you 1 mark. So I will just choose larger animals have a higher rate of respiration. Okay. Now, the respiration rate on the graph is measured in centimeter cubed of oxygen per minute. When the respiration rate is measured in centimeter cubed per minute, a human has a higher respiration rate than a mouse. When the respiration rate is measured in centimeter cubed per minute per gram of body mass, a human has a lower respiration rate than a mouse. Explain why a human has a lower respiration rate than a mouse when the rate of respiration is measured in centimeter cubed per minute per gram of body mass. Okay, so Number one is that humans have a smaller surface area to volume ratio when compared to the mouse. So if you get a mouse and you open it up but don't, if you get a mouse, the ratio of the size of his lungs is fairly larger than the ratio if you have a human being and his lungs. So humans have a smaller so humans have a smaller surface area to volume ratio. Your next question, since this is a question that's talking about respiration and breathing, we have to talk about heat loss. Okay? So, we can say that for a human requires less respiration to maintain body temperature. Since a mouse is smaller, it's gonna need to respire more to stay warmer. So, I don't know how to. Okay, so if you have a human and a mouse and you put them in Antarctica out of nowhere, you use Bluetooth, boom, human and mouse in Antarctica. Which one is gonna survive longer? The human is gonna survive longer. Why? Because his body produces more heat, okay, than a mouse. A mouse is going to need to keep respiring faster because since it's already small, since it's like very small, it's going to be losing heat much faster. Next question. Okay. So, this apparatus can be used to measure the respiration rate of a small animal. Explain how the apparatus should be used to obtain reliable data. Keyword is explain. They want you to explain. It's very important. Even if you've never seen this apparatus before, which you probably might have seen, or sometimes they might take an apparatus that you studied in class and they might maybe change it a bit, but generally the concept is the same. You have an animal over here that's respiring, respiration, they take in oxygen, they let out CO2. This colored thing is going to move down here. The soda lime, what does soda lime do? Soda lime absorbs the CO2 released. So that means that as they take in oxygen, this is going to move down over here. It's going to move in this direction. And this syringe provides O2. Okay, so number one, you are going to have to measure the distance in centimeters of the movement of the colored liquid. Our next point mainly is on time. So if you're in a lab, everything you do is based off of time. You need to be organized. There needs to be time. Time is important when carrying out experience. Okay? Sorry, when carrying out experiments. Okay. So you're gonna have to carry out this measurement at a known time. So you're gonna have to say carry out the measurement every 5 minutes for a duration of 30 minutes. Okay, now remember, in experiments, it's always important to repeat. So you'll repeat the experiment which increases the validity of results. Okay, so measure the distance in centimeter of the movement in colored liquid. That's 1. Over here, carry out the measurement every 5 minutes for duration. That's also another mark. And then over here, when you say repeat experiment, that's 1 mark. Honestly, you don't need to mention this part. However, when you do A levels, you will have to. So that's 3 marks. Okay. Some bacteria are pathogenic and cause infections. The diagram shows the structure of a pathogenic bacteria. Name the labeled part X. Okay, so let's just recap on some knowledge. Bacteria is in the prokaryotic family. So bacteria is a prokaryotic. Remember, prokaryotic, their main characteristics is they have no membrane-bound organelles. Okay, so name the labeled part X. Oh, this is a plasmid. Boom. Okay, so plasmids are not needed for the survival of a bacteria. However, plasmids do contain certain genes that are advantageous to the bacterium. So plasmids can contain resistance to antibiotics. Oh, and also prokaryotes or prokaryotic organisms do not have a nucleus. So no true nucleus. This is the flagellum. This is circular DNA. Okay. So, antibiotics can be used to cure some infections. Some bacteria are resistant to antibiotics. Oh, this resistance, as I said before, is in the plasmid. Explain how resistance to an antibiotic occurs and increases in a population of bacteria. So the first point is a mutation occurs giving resistance to antibiotics. And then the second part is the bacteria with the mutation will survive. Then the second question, the third part is that bacteria multiply and pass on the resistance gene. You can say gene, allele, DNA, but I prefer to just say gene. So where did I get my points? Number one, by acknowledging that a mutation occurs, giving resistance to antibiotics. Number two, the bacteria with the mutation survive. It's important to state these main keywords: mutation, survive, and then gene. So bacteria, antibiotics, mutation occurs, some of them survive, they multiply, they pass the gene. Okay? So, okay, this is actually an interesting thing, how bacteria multiply. Bacteria multiply through something called binary fission. You don't need to know this. They're not going to specifically ask you about this, but however, they can pass their genes like by sticking next to each other. They can just pass their genes and DNA information. This is why antibiotics are not. This is why there's an attack on antibiotics where doctors don't want many people to start using them because they don't want bacteria to gain this advantage, or else it'll be very difficult to treat diseases and stuff like that. Okay. The table shows the number of deaths in 2014 caused by bacteria that are resistant to antibiotics. The table also shows the predicted number of deaths in 2050 caused by resistant bacteria. Okay, so this is times 10 to the power of 6, so this is millions. So in 2014, it's 0.7 times 10 to the power of 6. 2050, it's 10 times 10 to the power of 6. Calculate the percentage increase in predicted number of deaths in 2050. Compare with the number of deaths in 2014. Okay, so this is our original. Remember that our original is going to go in our denominator. So it's going to go over here. Then we're going to have the times 100. Okay. So 0.7 times 10 to the power of 6 minus, wait, 1 second. You need to minus the larger number from the smaller one. Brackets minus 0.7 times 10 to the power of 6. Always write out your calculations. It's very important to write out your calculations. I remember when I was doing my GCSEs and A-levels, all my teachers were like, Omar, you have to write out your calculations. Honestly, I used to write them all the time because I just liked how neat it looked, and it's very important that you get marks when you write out your calculations. Okay, so this is my answer: 9, 3, 1, 2, 3, 4, 5. Okay, 1, 2, 3, 4, 5. 93. What is that? Oh, that's 9,300,000. Okay. Now we're gonna have 9,300,000 over our original value times 100. Let's put that in a calculator. And we get a whopping increase of 1,300 and 1,328.57%. Okay, so you're gonna have to round this up. Since this is a 5, this pushes this one up, so you'll just say 1,329%. It's very important to put the percentage sign. Okay. Okay, next question. A doctor claims that if he stops giving antibiotics to any patients who are ill, he can reduce the number of predicted deaths caused by resistance bacteria in 2050. So, remember that we talked about that giving antibiotics increases the amount Okay, so giving antibiotics increases the amount of resistant bacteria. Therefore, decreasing the selection pressure on bacteria. So decreasing selection pressure on bacteria basically means that there's less stuff that is gonna kill the bacteria and get rid of it, meaning that bacteria can just grow and grow. Okay, so this means that antibiotics will become less effective. Okay, this means that we need to relate back to the question of a doctor. So I will just mention that patients will die as antibiotics are not working. Okay, now a student is investigating the effect of temperature on gas production in yeast. Yeast is a fungi. They have chysin. Okay. The student uses this apparatus to count the number of gas bubbles produced per minute. The gas, since yeast is fermenting, it will produce CO2. Okay, at different temperatures. Name the process in yeast that produces the gas bubbles. So, gas bubbles are produced by fermentation. You can also mention respiration, and it's also correct, but I prefer to stick to fermentation. Okay. What gas is produced by the yeast? CO2. This is really important in... Okay, so basically what fermentation happens, it is the recycling of NADH to make NAD+, which is needed for glycolysis. This will be further explored in your A-levels. Okay, plot a graph to show these results. Join the points with a straight line. Okay, this is going to be difficult to draw on a computer, but I'll try my best. Okay, so with a graph, it's really important to label. Okay, very important to label. Make sure that you label. Your labels must be drawn in pen, and then the line must be drawn in pencil. Okay, that's very important. So I'll put my temperature on the x-axis. Whatever is drawn here, whatever is written here, just make sure you write it exactly the same down there. and on the y-axis. So I will just say number of bubbles per minute. Okay, obviously this needs to be much more neater than this, but hopefully you guys get the idea. Okay, so it goes in 20, 25, 30, 35. Okay. Let's see if this works. Okay, so 20, 25, 30, 35, 40, 45, 50, 55. Okay. Your graph to get 1 mark, at least 1 mark, it needs to cover at least 50% of the given space. So I can just write 20, 25, 30, 35, 40, 45, 50, 55. Okay. And then over here, our highest number is 20. So I can say 1, 2, 3. Okay, I can do 5, 10, 15, 20. Nope, that's not possible, because if you do it in 0, 5, 10, 15, 20, it will barely covers 20% of the graph. So maybe aim to be around here. Okay, so we can do it 2, 4, 6, 8, 10. Yes, we can. So here, 0, 2, 4, 6, 8, 10, 12, 14. That's a 4. 16, 18, 20, 22. Okay, this is a 4, but it's a messed up 4. Okay, so at 20 degrees, we have 2 bubbles. Make sure that you draw your points in X. Don't do dots. Don't do multiple things like that. Needs to be just an X. Okay. Boom. 25, 6. So 6. Boom. 30 is 8. Okay. 35 is 10. 40 is 14. 45 is 20. 50, no data. 55 is 2. Okay. And then you're going to have to draw a line of best fit. Okay. Obviously, I can't put a ruler on top of my laptop. I'll just try to. Hopefully this works. Okay. Okay. Well, generally speaking, your line must cover the most amount of points. So technically, it should move a little bit down there, but it's difficult to do that while on a laptop. But this will definitely get you marks. Okay. Explain the result at 55 degrees Celsius. So if we notice that at 55 degrees Celsius, it suddenly just goes all the way down. Remember that enzymes are proteins, and proteins have bonds, chemical bonds, which are affected by temperature. So at 55 degrees Celsius, all you have to write is number 1, enzymes denature. And then you will say that since it goes up in high temperature, this causes bonds to break, causes the proteins, because proteins make up an enzyme, proteins will unfold, which changes the enzyme's active site. So you'll just say change in shape of active sites. Perfecto. Okay. Describe how Describe how the student should modify the investigation to obtain a more accurate measure of optimum temperature. Okay. So if you guys look back to the graph, there is a huge gap between 40 and 45. So first of all, he should do it at smaller intervals. So read / measure at smaller intervals of temperature. Between 40 and 45. Because if I could go back, you will see that we have a graph like this, and then suddenly over here we have at 45 and at 50, it just goes all the way down here. Now we have this huge area over here that we have no data about, so at smaller time intervals it will allow us to see the small changes. Usually it's like this, so it's going to be like this. That's how the graph of the enzyme usually goes. Over here I showed you this beautiful graph over here. So this is the optimum temperature where the enzyme is most active. Okay. Okay, so the diagram shows some energy transfers along a food chain. The numbers give the energy transfers in kilojoules per meter cubed per year. So give the name of the process X. Okay, we have producers, primary consumers, secondary consumers, tertiary consumers, or decomposers, and process X. Okay, so process X, remember, decomposers and all organisms in this world will respire, so this process is respiration. You can also say heat loss. However, I prefer that you stick to respiration, because in the process of respiration, heat loss occurs. It's simultaneous. Okay, determine the energy transferred from the primary consumer to the secondary consumer. Okay, so we have our consumer over here, and we have energy loss over here, and this is going to go up to our secondary consumer. Okay, so just these arrows indicate the loss. So this is just loss. This is our gain. So our gain is 10,200, and our loss is 2,040 and 7,038. Okay, so this is very simple. We just want to know what's going to go up here. This is lost. This is our gained. Okay, so we're going to do a simple calculation. Minus equals 1,122. Okay. Decomposers can be described as saprotrophs. Describe how saprotrophs cause decomposition. Okay. So decomposers, they will break down material. So just write break down And then our second point is they will secrete enzymes to help this breaking down process. So secrete enzymes to aid in decomposition. Okay. 40% of the energy in the producers transfers to decomposers. Only 20% of the energy the primary consumers transfers to decomposers. Explain this difference in energy. Okay, so plants are made up of cellulose, and cellulose is very difficult to digest. So plants contain cellulose, which is very difficult for decomposers to digest. And then also, you can say primary consumers lose a lot of energy in respiration. Energy is lost in the form of heat. Yeah, but this and this are very good points. Over here, you said that plants contain cellulose. That's one point, which is very difficult for decomposers to digest. That's another point. And then primary consumers lose a loss of heat and energy. That's the third point. Okay. Now, different factors can affect the rate of photosynthesis in plants. Give the balanced chemical equation for the process of photosynthesis. On this equation, you just have to memorize it. Like, you have to memorize it and understand it, but honestly, it's all about memorization. And if you take chemistry GCSE, you will understand the concept of balancing equations. So it's 6 carbons plus 6 water will give you C6H12O6 plus 6O2. This is our glucose. This is our oxygen. Obviously, plants need carbon and water. Okay. A student uses this apparatus to investigate the need for carbon dioxide in photosynthesis. Over here we have a leaf. Okay, we have a leaf over here that is in a flask with some sodium hydroxide. Sodium hydroxide absorbs CO2. Okay, explain how the student could use this apparatus to show that carbon dioxide is needed for photosynthesis. Okay, so photosynthesis needs light. Okay, needs light and carbon dioxide to make glucose. How do we test for glucose and starch? Well, we test for starch using iodine. So keep that point in mind because this is going to be needed for a future question. So number 1 is we have to place the plant in a dark area for 24 hours to remove any starch present. And then you're going to have to place the sodium hydroxide with the plant in the dark to remove CO2. And then you're going to have to expose. No, wait, let's do that from the beginning. Wait, I forgot to mention a point. Honestly, this question is a bit difficult because some students have a difficult time to understand, to relate concepts together. However, in all past papers and all questions in all exams, there's always going to be this one question. Okay, so number 1 is place the plant in the dark for 24 hours to de-starch it. And then put 1 leaf in a flask with sodium hydroxide to remove CO2. And then you'll expose the plant to light. Then test this leaf and a control leaf using iodine solution. So technically only the control leaf will highlight a blue-black color. Okay, this is a grade 9 question. This is a grade 9 question. Okay. The student is told that after keeping the leaf in the flask for a day, he should cut the leaf into small shapes for testing. Suggest why this is a good idea. Okay, so this is just asking about your this is asking about ethical ways of carrying out experiments and stuff like that. And also this requires some critical thinking. So when you have 1 leaf and you cut it into multiple pieces, you're gonna be using fewer plants. So fewer plants needed. And then also, since you have many smaller pieces, you can have multiple repetitions. Okay, perfect. The results of this investigation show whether carbon dioxide is needed for photosynthesis. Explain how the student could modify this investigation to show that chlorophyll is needed for photosynthesis. Actually, in one of my previous labs, we did this experiment where we got two geranium leaves. We boiled both of them. However, one of them we dipped in ethanol. Now, ethanol is gonna remove all the chlorophyll, okay? It removes all the chlorophyll and every starch and everything. And then our leaf that has chlorophyll present, when we put iodine on it, it gave us a blue-black color indicating the presence of starch. The one where we removed the chlorophyll, it gave us no color because there was no chlorophyll, meaning that there was no starch. So over here, we can use a variegated leaf, or you can explain the whole boiling geranium thing. However, variegated leaf is usually the way to go. Okay, so number 1 is use a variegated Now the variegated leaf has a mutation where this white part has no chlorophyll and this green part has chlorophyll. And then you will compare. Use a variegated leaf and compare the color when iodine solution is added. Oh, you also need to do one more thing. Okay, this should be the first stop over here. You must keep all leaves in the same conditions. Okay. Next question is about the eye. The diagram shows a section through the human eye. Name the structures labeled A, B, C, and D. Okay, when it comes to the human eye and the heart, you need to practice, you need to keep drawing them out. You need to keep, you need to have a drawing and put it in front of your test. That's how I memorized it. And I just kept, every day I was just drawing one eye and then labeling it. Okay, so A is our iris. Now B, okay, B over here is our cornea. And C is our pupil. And D is our lens. Okay. Okay. When a person looks directly at an object, light is focused on the foeva of the retina. A person is reading a book and then looks at a distant object. Explain the changes that occur in the structure of the eye that allow light from a distant object to be focused on the foeva. Okay, so just please, when you're reading this question, do not confuse. Like, look over here how they said looks directly at an object, okay? And then the actual question is on a distant object. Just notice how the questions and how they word it so you don't get confused. Okay, so looking at a distant object. So the ciliary muscles relax, causing the suspensory ligaments causing the suspensory ligaments to pull tight. Oh, they also might use the word taut. Yeah, I think I spelled it wrong. Or just taut. But yeah, I just prefer to write pull tight. Okay, this makes the lens thinner. And less curved. So light is refracted less. When you're looking at a distant object, your pupils will dilate. Dilate becomes bigger. Okay. Some people develop a condition called age-related macular degeneration, AMD. This occurs when cells in the foeva of the retina are damaged. Suggest how AMD affects vision. So you're going to have loss of vision. And also you can say loss of color. Okay. Scientists are developing a new treatment for AMD. The treatment involves an operation to replace the damaged cells in the foeva with new cells. Early results show an improvement in the condition. However, the treatment has only been tested on 2 patients. Suggest what else scientists need to do before the treatment is allowed to be used for more patients. Number 1, they need to, since they said they mentioned a number over here, only 2, so that number is not sufficient. So repeat with more patients. Usually in scientific studies and medical studies, they will carry this experiment using like at least 1,000 people. And then if this study has just been conducted for 2 weeks, it's unreliable. So the study needs to be studied for more years and for a longer process, and they need to monitor the progression of the patients. So just mention longer study, longer duration conducting the study. Okay. Question 9. Syndactyl is a condition where fingers and toes are not separated during development. The photograph shows the foot of someone with syndactyl. One type of syndactyl is caused by a dominant allele that is inherited from the parents. A man without syndactyl and a woman with syndactyl have a male child. The child does not have syndactyl. Use a genetic diagram to show genotypes of the parents, the possible gametes, and the genotype and phenotype of the child. Use D to represent the dominant allele and D to represent the recessive allele. Okay, so okay, so the first thing we have to do is we need to write the father over here. Father, mother, and then we have to write genotype And then I like to write their genotypes, their gametes, and then phenotypes of child. Remember, phenotype is the characteristic that is expressed. Gametes are usually sexual cells. Okay, so this condition is caused by a dominant allele. Okay, so first of all, let's just have our father. Males have XY chromosomes, XY. Females have XX. Okay, and then let's use green. Okay, so this condition is caused by a dominant allele. Okay, however, the child does not have it. So that means that it is not dominant in the mother. It's going to be one dominant and one recessive. So we are going to write capital D and then a small d, recessive, dominant. And then for the father, does the father carry it? Let me just read the question again. Okay, a man without syndicate and a woman with syndicate have a child. The child does not have syndicate. Syndactyle. What am I saying? Syndicate. Okay. Okay. So the father doesn't have it. So that means that the father is recessive, small d. Okay. And then we are gonna have our, okay, so it's x d y, x d d. You're gonna do your Punnett square. Let's put our male over here. X d y. Then we're gonna have, let's just put d over here, d over here. Now our mother, who has it? Oh, let's use red so that you guys can understand this better. Okay, x dominant d, x small d. Okay, boom, boom. Okay, so this is gonna be x d y. Oh no, our tables, they're broken. Wait, let's just do this better so it's cleaner. Okay, y over here, y over here, and then x d. Okay. We're gonna have xd capital. XD is capital over here, XD over here, so dominant. It's going to be a small XD, so nothing over here. The child is okay. And then this is going to be XD. This is also going to be XD. Okay, female, male. Okay, this is our female, male, male. Okay, yeah, this is what we have over here. Okay, so we have XD dominant and XD recessive. Okay, dominant, recessive. Then we're going to have XD dominant, YD recessive. Okay, then we're going to have X recessive, and then X recessive, Y recessive. Okay. Okay, determine the probability that the second child of the parents will be a girl with syndicate. So the probability of being a girl is 50%, and it's going to be 0.5. So 0.5 for being a girl. And then the probability of having the condition is also going to be 0.5. 0.5 times 0.5 equals 0.25, which is times 100, which gives us 25%. Okay. Okay. Another type of syndicate is caused by a recessive allele. Recessive. So that's small d, small d. Okay. Explain the difference in the pattern of inheritance between a condition caused by a dominant allele, okay, a dominant allele caused by a recessive allele, okay. So this is a form of polygenetic, so it's going to be polygenetic. Then there's obviously more than one gene affecting the phenotype. There's more than one gene affecting and then our small one is each recessive allele has a small effect. Just each allele has a small effect. This is also a grade 9 level question. Okay, next question. Okay, syndactyl is controlled by a single gene that has two alleles. Most other phenotypes are the result of a gene sort of genetic control. Describe the genetic control of most phenotypic features. So it is going to be polygenetic. Polygenic, sorry. And then there's going to be many genes control one phenotype. Also, I would like to mention that the previous question that I wrote was answering this question over here. So that's my apologies. The previous question back there is wrong because I answered it wrong. This is the correct one. Okay. Question 10. The body mass index BMI is a measure that uses your mass and height to calculate if your mass is appropriate for your height. BMI is calculated using the formula mass in kg times height in meters squared, and then to the power of 2. Person X has a mass of 60 kg and a height of 165 centimeters. Calculate the BMI of a person. Okay, so from centimeters to meters, you're going to have to divide it by 100. Just note that. Okay, so BMI, mass in kg, so 60 divided by 1.65 squared, okay, equals 22. Okay, 22. Okay, BMI values can be divided into categories. The table shows these categories. Give the category of person X. Okay, so 22 lies within over here, so he is healthy. Okay. One problem with the interpretation of BMI measurements is that people have different proportions of muscle compared with fat. Muscle has a higher density than fat. What would what would a high portion of muscle in your body have on your BMI? Muscle will make your fat higher, and in calculating BMI, remember you are going to multiply kg. So it's kg by your height. So your BMI, it would cause an increase. Okay. Some diets reduce carbohydrate intake and other diets reduce lipid intake. Explain how these diets can result in a reduction of body mass. Okay, so carbohydrates and lipids are high in energy. Okay, so energy intake is going to be less than the energy used. This means that your body will use the stored fat, and it will be respired. Your body will burn the stored fat during respiration. Okay. Explain why diets are more effective at reducing BMI if combined with regular exercise. What does exercise do? Exercise causes muscle contraction. So muscle contraction requires energy. Okay, this energy is a result of respiration. So more energy is being lost as you respire more. Exercising causes you to respire more, therefore losing more fat faster. Okay, question 11. Give the name of this cycle. So we have CO2 in the atmosphere, we have plants, we have animals, we have gases going in the soil, soil microorganisms. We have a factory over here releasing gas, CO2. So this is the carbon cycle. Okay. Which process reduces CO2 in the atmosphere? Reduces CO2. I wonder which one it is. Oh, look, it's A, plants, photosynthesis. Name two groups of microorganisms that carry out process B. So process B is decomposition. So the organisms are bacteria and things like mushrooms, so fungi. This is 1, this is 2. Okay, now this is our final question where we're going to be using CORMS. So CORMMS. Change, organism, repeat, measurement, and the same. So this acronym is very important when answering these type of questions. Okay, the breakdown of dead plant material by microorganisms is affected by many factors. Design an investigation to determine the temperature at which plant material is broken down most effectively. Include experimental detail in your answer and write in full sentences. Okay, so first of all, we are going to set up containers of dead plant material at 20 degrees Celsius and 40 degrees. It's very important to state the temperature because temperature is one of the variables. And then I must use the same species of plant material. And you also must use the same type of organism. So, and the same type of bacteria. Because bacteria is our organism that does the decomposition. Okay, then it's very important to, so CO corms. It's very important to mention repeat. Okay, then I will repeat. Then I will repeat the experiment for 6 times. You don't have to say how many times, but I like to say 6 times. At each temperature. You need to specify what exactly you're going to do. So at each temperature. This ensures the results are reliable. And then I would also measure the amount of carbon dioxide being released as an indicator of decomposition. Other variables such as moisture and oxygen availability must remain constant. Okay, so here you set 6 points, which should get you 6 marks. Thank you so much for watching this video, and I hope that I helped you in some way, in some form. And if you would like me to do any more past papers, just write down the dates in the comment section, and I will hopefully do them as fast as possible. Also, if you'd like me to explain any topics or tell you how I studied and give you tips and tricks, just let me know. Thank you so much.